Hello everyone. Today we are going to discuss about an important topic in biochemistry which is related to the biomolecule carbohydrate, and that is the Benedict's test. So Benedict's test, the overall significance of the Benedict's test is to detect the presence of reducing sugar in a particular sample. We will discuss about in detail. Yes. Coming to the Benedict's test, it is the biochemical test which used to detect the presence of reducing sugar in a sample, and that sample may be of urine, carbohydrate or any type of carbohydrate solution like glucose, fructose, etc. And this Benedict's test is most commonly applied in the clinical diagnosis. So, where in clinical diagnosis is to detect the presence of glucose in urine for the screening of diabetes mellitus. So in sugar patients, the glucose is elevated in the urine, and in that urine we used to detect the reducing sugar of glucose is present or not. And it is also involved in the food analysis, that is how sugar content in several products have been identified, such as in cakes and biscuit and some of the desserts containing sugar content, and to analyze that sugar content in some of the dairy products or food products, it can be used in food analysis also, and in the purpose of educational laboratories. So as I said before, it comes under biomolecule carbohydrates. So carbohydrate identification purpose in educational laboratories, this Benedict's test have been used widely. As coming to the carbohydrate properties, this Benedict's test is mostly comes under reducing sugar property. So in carbohydrates there are mainly two properties. First one is physical property and second one is chemical property. In physical property, the structural variation, the structural similarities have been discussed, and in chemical properties this Benedict's test is involved in the reducing property of sugar. And reducing property of sugar, it can donate electrons to reduce other compounds. Yes, it can donate the H plus electrons to reduce other compounds, so that it reduces the properties. So for example, some of the reducing sugars from monosaccharides are glucose, fructose and galactose, and disaccharides maltose and lactose. So you could see the image of glucose, mannose and fructose there. In glucose, the aldehyde functional group is present. In mannose also aldehyde functional group. Aldehyde functional group is CHO, and this CHO is indicated in the aldose form. And whereas in fructose, ketone exists as functional group. Ketone is C double bond O. So the reducing sugar have the free aldehyde or free ketone functional group in their structure. So this is the overall structure of the reducing sugar property of carbohydrate. First one is D-glucose, which these three having the same structural chemical formula of C6H12O6, where you could find C carbon atom 6, hydrogen atom 12 and oxygen atom 6. And only the functional group will get vary from glucose and fructose. Glucose having aldehyde functional group, that is aldose, and fructose having ketone functional group, that is ketone. And coming to the materials required, there are these following materials are required for the carrying out of Benedict's test. First one is Benedict reagent, and second one is test sample, that is urine sample or carbohydrate solution or any other, and test tubes. So for this test we need test tubes of any size, and most importantly is water bath or Bunsen burner for heating that solution. So and dropper or pipette. So you must add sample and Benedict's reagents in this dropper or pipette, and finally the distilled water. And this distilled water is for control purpose, where you could compare our result with our control. And Benedict's reagent comprises of three solutions, that is copper sulfate and sodium citrate and sodium carbonate, where Benedict's reagents appears in the form of blue color, and this blue color formation is due to these three chemicals, that is copper sulfate, and sodium citrate and sodium carbonate, and these three liquid together forms the Benedict's reagent solution, and it can be ready-madely available in the market, or by using these three reagents we can prepare the Benedict solution.